Hello students, today we're going to look at microevolution. We'll look at 4 key core concepts. In concept number 1, we'll look at genetic variation, how it makes evolution possible. In key concept number 2, we'll look at the Hardy-Weinberg equation, how it can be used to test whether a population is evolving. In key concept number 3, we'll look at natural selection, genetic drift, and gene flow, and how it can alter allele frequencies in a population. And finally, in key concept number 4, we'll look at natural selection, the only mechanism that consistently causes adaptive evolution. So let's have a look at the smallest units of evolution. One common misconception about evolution is that individual organisms evolve. It is true that natural selection acts on individuals. Each organism's traits affect its survival and reproductive success compared with those of other individuals. But the evolutionary impact of natural selection is only apparent in how a population of organisms changes over time. For example, consider the medium ground finch, G. fortis, a seed-eating bird that inhabits Galapagos Islands. In 1977, G. fortis' population on the island of Daphne Major was decimated by a long period of drought. Of some 1,200 birds, only 180 survived. Researchers Peter and Rosemary Grant observed that during the drought, small soft seeds were in short supply. The finches mostly fed on large hard seeds that were plentiful. Birds with larger, deeper beaks were better able to crack and eat these larger seeds, and they survived at a higher rate than did finches with smaller beaks. Since beak depth is an inherited trait in these birds, the offspring of surviving birds also tended to have deep beaks. As a result, the average beak depth in the next generation of G. fortis was greater than it had been in the pre-drought population. So the figure shows the finch population had evolved by natural selection. However, the individual finches did not evolve. Each bird had a beak of a particular size, which did not grow larger during the drought. Rather, the population of large beaks in the population increased from generation to generation. The population evolved, not its individual members. Evolution on its smallest scale is called microevolution. This is a change in allele frequencies in a population over generations. And as you will see later in the lecture, natural selection is not the only cause of microevolution. In fact, there are 3 main mechanisms that cause allele frequency change: natural selection, genetic drift, these are chance events that alter allele frequencies, and gene flow, the transfer of alleles between populations. Each of these mechanisms has distinctive effects on the genetic composition of populations. However, only natural selection consistently improves the degree to which organisms are well suited for life in their environment. Adaptation. Before we examine natural selection and adaptation more closely, let's revisit a prerequisite for these processes in a population: genetic variation. In The Origin of Species, Darwin provided abundant evidence that life on Earth has evolved over time, and he proposed natural selection as a primary mechanism for that change. He observed that individuals differ in their inherited traits and that selection acts on such differences, leading to evolutionary change. Although Darwin realized that variation in hereditable traits is a prerequisite for evolution, he did not know precisely how organisms pass hereditable traits to their offspring. Just a few years after Darwin published The Origin of Species, Mendel produced a model of inheritance in which organisms transmit discrete hereditable units, now called genes, to their offspring. Although Darwin did not know about genes, Mendel's paper set the stage for understanding the genetic differences on which evolution is based. We will now look at those differences. So let's have a look at genetic variation. Individuals within all species vary in their phenotypic traits. Among humans, for example, you can easily observe phenotypic variation in facial features, height, and voice. Indeed, individual variation occurs in all species. And though you cannot identify a person's blood group, whether it's A, B, or O, from his or her appearance, this and many other molecular traits also vary extensively among individuals. Such phenotypic variations often reflect genetic variation, differences among individuals in the composition of their genes or DNA sequences. Some heritable phenotypic differences occur on an either-or basis, such as the flower colours of Mendel's pea plants, Each plant had flowers that were either purple or white, if you can remember. Characters that vary in this way are typically determined by a single gene locus, with different alleles producing distinct phenotypes. In contrast, other phenotypic differences vary in gradations along a continuum. Such variation usually results from the influence of two or more genes on a single phenotypic character. In fact, many of phenotypic characters are influenced by multiple genes, including coat color in horses, seed number in maize, and height in humans. So, how much do genes and other DNA sequences vary from one individual to another? Genetic variation at the whole gene level, gene variability, can be quantified as the average percentage of loci that are heterozygous. Recall that heterozygous individuals has two different alleles for a given locus, whereas a homozygous individual has two identical alleles for that locus. A good example is the average fruit fly, Drosophila melanogaster, which is heterozygous for about 1,920 of its 13,700 loci, 14%, and homozygous for all the rest. Considerable genetic variation can also be measured at the molecular level of DNA, nucleotide variability, but little of this variation results in phenotypic variation. Why, might you ask? Many nucleotide variations occur within introns, segments of DNA lying between exons, the regions retained in mRNA after RNA processing. And of the variations that occur within exons, most do not cause change in the amino acid sequence of the protein encoded by the gene. For example, in the sequence comparison shown in the next slide, there are 43 nucleotide sites with variable base pairs where substitutions have occurred, as well as several sites where insertions or deletions have occurred. Although 18 variable sites occur within the 4 exons of the ADH gene, only one of these variations at site 1490 result in an amino acid change. Note, however, that this is a single variable site, and this is enough to cause genetic variation at the level of the gene, and hence two different forms of the ADH enzyme are produced. It's important to bear in mind that some phenotypic variation does not result from genetic differences among individuals. Phenotype is the product of an inherited genotype and many environmental influences. In a human body, for example, bodybuilders alter their phenotypes dramatically but do not pass their huge muscles on to the next generation. In general, only the genetically determined part of phenotypic variation can have evolutionary consequences. As such, genetic variation provides a raw material for evolutionary change. Without genetic variation, evolution cannot occur. So let's have a quick look at sources of genetic variation. The genetic variation on which evolution depends originates when mutation, gene duplication, or other processes produce new alleles and new genes. Genetic variants can be produced rapidly in organisms with short generation times. Sexual reproduction can also result in genetic variation as existing genes are arranged in new ways. So let's have a look at the formation of new alleles. New alleles can arise by mutation, a change in the nucleotide sequence of an organism's DNA. A change as little as one base in a gene, a point mutation, can have significant impact on phenotype, as in sickle cell disease. We might expect that this would be the case. Organisms reflect many generations of past selection, and hence their phenotypes tend to be suited for life in their environments. As a result, most new mutations that alter a phenotype are at least slightly harmful. In some cases, natural selection quickly removes such harmful alleles. In diploid organisms, however, harmful alleles that are recessive can be hidden from selection. Indeed, a harmful recessive allele can persist for generations by propagation in heterozygous individuals, whereas harmful effects can be masked by the more favorable dominant allele. Such heterozygote protection maintains a huge pool of alleles that might not be favored under present conditions, but that could be beneficial if the environment changes. While many mutations are harmful, many others are not. Recall that much of the DNA in eukaryotic genomes do not encode proteins. Point mutations in these non-coding regions generally result in neutral variation, differences in DNA sequence that do not confer a selective advantage or disadvantage. The redundancy in the genetic code is another source of neutral variation. Even a point mutation in a gene that encodes a protein will have no effect on the protein's function if the amino acid composition is not changed. And even when there is a change in the amino acid, it may not affect the protein's shape and function. Finally, note that in multicellular organisms, only mutations in cell lines that produce gametes can be passed to offspring. In plants and fungi, this is not as limiting as it may sound, since many different cell lines can produce gametes. However, in most animals, the majority of mutations occur in somatic cells and are not passed to offspring. So let's have a look at altering gene number or position. Chromosomal changes that delete, disrupt or rearrange many loci are usually harmful. However, when such large-scale changes leave genes intact, they may not affect the organism's phenotype. In rare cases, chromosomal rearrangements may even be beneficial. For example, the translocation of part of one chromosome to a different chromosome could link genes in a way that produces a positive effect. A key potential source of variation is the duplication of genes due to errors in meiosis, such as unequal crossing over, slippage during DNA replication, or the activity of transposable elements. Duplications of large chromosomal segments, like other chromosomal aberrations, are often harmful, but the duplication of smaller pieces of DNA may not be. Gene duplications that do not have severe effects can persist over generations, allowing mutations to accumulate. The result is an expanded genome with new genes that may take on new functions. Such increases in gene number appear to have played a major role in evolution. For example, the remote ancestors of mammals have a single gene for detecting odors that has since been duplicated many times. As a result, humans today have about 380 functional olfactory receptor genes, and mice have about 1200. This dramatic proliferation of olfactory genes probably helped early mammals, enabling them to detect faint odors and to distinguish among many different smells. So let's have a look at rapid reproduction. Mutation rates tend to be low in plants and animals, averaging about one mutation in every 100,000 genes per generation, and they are often even lower in prokaryotes. But prokaryotes have many more generations per unit of time, so mutations can quickly generate genetic variation in their populations. The same is true of viruses. For instance, HIV has a generation time of about 2 days. That is, it takes 2 days for a newly formed virus to produce the next generation of viruses. HIV also has an RNA genome, which has a much higher mutational rate than a typical DNA genome because of the lack of RNA repair mechanisms in host cells. For this reason, single drug treatments are unlikely to be effective against HIV. Mutant forms of the virus that are resistant to a particular drug would tend to proliferate in relatively short order. The most effective AIDS treatment to date have been drug cocktails that combine several medications. This approach has worked well because it is less likely that a set of mutations that together confer resistance to all the drugs will occur in a short time period. So let's have a quick look at sexual reproduction. In organisms that reproduce sexually, most of the genetic variation in the population results from the unique combination of alleles that each individual receives from its parents. I think we know that. Of course, at the nucleotide level, all the differences among these alleles have originated from past mutations. Sexual reproduction then shuffles existing alleles and deals them at random to produce individual genotypes. 3 mechanisms contribute to this shuffling: crossing over, independent assortment of chromosomes, and fertilization. Now, during meiosis, homologous chromosomes once inherited from each parent trade some of their alleles by crossing over. These homologous chromosomes and the alleles they carry are then distributed at random into gametes. Then, because myriad possible mating combinations exist in the population, fertilization typically brings together gametes that have different genetic backgrounds. The combined effects of these 3 mechanisms ensure that sexual reproduction rearranges existing alleles into fresh combinations each generation, providing much of the genetic variation that makes evolution possible. So that is the end of key concept number 1.